Alright guys, the third and final video here in the series that describes the phases of cell respiration is going to get into detail about the electron transport chain and the process known as oxidative phosphorylation. Okay, so the previous two videos in this series dealt with the first phase known as, there it is, glycolysis, and the second one dealt with the Krebs cycle, and finally the payoff, the big payoff here, is the electron transport chain, where we get a majority of our ATP from. Okay, so far up until this point, I think I ended the last video kind of emphasizing this. The ATP that we have generated has been by a process known as substrate level phosphorylation. Okay, and so this is the two net ATPs we made in glycolysis and the two ATPs we made in the Krebs cycle. They were made by this process with the enzyme called substrate level phosphorylation. A bulk majority of the ATP that we make from here on out in the electron transport chain, or I should say at the electron transport chain, is made through a process known as oxidative phosphorylation, also known as chemiosmosis. Okay, so chemiosmosis is the process that makes these ATP molecules. The structure that helps this process occur is known as the electron transport chain. Oftentimes those two things get confused. So the electron transport chain is a series of electron carrier molecules. These are molecules that will interact with high energy electrons in a different way. And what they do is they transfer the electrons to each other in kind of a sequence through a series of redox reactions. And through each one of these little reactions that these electrons work their way through in the chain, they will release small amounts of free energy. Okay, so this electron transport chain is located within the inner mitochondria, or inner membrane of the mitochondria, and what it does is it transfers of electrons through the membrane allows for the mitochondria to set up something known as a proton gradient, or a gradient of H+. And that gradient of H+ is actually what helps drive ATP production. And you would be absolutely mind blown, and you will when hopefully when you see the video, of how this process works, how these little H+ protons are actually utilized to spin like a little molecular turbine, almost identical in fashion to how they would make energy at the Hoover Dam. Okay, where water goes through this machine and spins a turbine and you get energy from it. There is something absolutely similar to that in this process, and so we're going to get into that a little bit as well, because I think it's just awesome. Without this electron transport chain, that transfer of the high energy electrons from hydrogen to oxygen is really, really explosive, and I think we've seen that before. So let's get into it. I'm gonna look at these two diagrams right here. And I'm going to mark up on all of them so that you can kind of get an idea of what I just talked about here. Okay, so I'm going to escape the presentation and pop this guy up right here. All right, so what you see, I'm going to try to place myself maybe right here, right up here. Okay, that's a good spot. What you see here are two different diagrams. This first diagram shows all of these different membrane proteins, okay, these electron transport chain proteins. That's what these guys all are, these purple protein things. And those are the same things that you would see over here, okay? That's those. They are just placed in a different diagram is all, okay? Now, let's try to make heads and tails of what this diagram on the left is. This diagram on the left shows us these proteins in the electron transport chain against a free energy, a scale of free energy, okay? And it shows us this very first set of proteins in the complex, kind of right up here. That would correlate to this one right here, okay? Now, I'm going to use a different color. Can I? Eraser? Yeah, there we go. Get the eraser, see if I can use a different color. Maybe blue. I like that blue. Okay. This one here correlates to this one here. All right. So that's where we are. And we're starting there because that's where NADH, as we see here, and also here, drops off those high energy electrons first. Okay, so NADH is going to bring those high energy electrons. Remember those things, those NADHs are now electron carriers, electron transporters, right? They're going to bring those high energy electrons to these proteins in the electron transport chain and just drop them off. Okay? And those electrons are dropped off with an associated energy of about 40. That's hard to see. Let me see if I can erase again. There we go. All right. An associated energy, those electrons are dropped off with an associated energy of roughly 40 kilocalories per mole. That's what we see here. So they've got some energy, right? And that's a relatively high amount of energy. And what those electrons are going to do is they are going to bounce through each of these little proteins, and in interaction with each one of them, they're going to undergo some kind of redox reaction all the way through. So as they bounce through each one of these little proteins in sequence, like we kind of see over here as well, they will have some kind of chemical interaction, okay? And ultimately, those electrons kind of end up all the way down here. So they make it from this protein here all the way through all of these proteins down to this one here, number 4 in sequence, all right? And what you notice is that once they get down to this one, or to the very last one, there's far less energy associated with them. We'll call this like 50 kilocalories per mole. There's far less energy associated with those electrons than when they started at 40 kilocalories per mole. Okay, so what do we need to think about in terms of what I just said? When those electrons went through all of these little chemical interactions or chemical reactions, they started with 40 kilocalories per mole and they ended with 15 kilocalories per mole. What you could say about those electrons is that they lost energy. They lost free energy, right? And what can we say about free energy that is lost? We can say that it is harnessed to do work, okay? And that's effectively what we see, okay? This energy right here is harnessed to do some work. Now let me see if I can erase. Yes, I want to erase. I want to erase all this baloney here. So we've got some free energy to use, right? We've got some capital here. I'm going to put it off to the side so we know that we can spend it, right? Now let me draw a little bit something else here. Let's say this is the mitochondrial membrane. Okay, those are the borders of the mitochondrial membrane. What I just drew in red. Okay, so that would correlate to like this is the inner side, this is the inner side, this is the outer side, this is the outer side. Okay, now because these electrons have worked their way through these proteins and released free energy, we now have some energy to spend. Okay, and what we are going to do, or what the mitochondria I should say is going to do, is they are going to utilize H+. There's a buildup of H+ in here, through various aspects. And if you go back and you trace some of the things that we've learned even in this chapter, you'll see little random H+s all over the place. And they kind of build up in the inner mitochondrial matrix, which is right here where I put this red squiggly line. And that correlates to what you see right here. Okay? And now these H+s are going to be pumped to this side. Okay? They're going to be sent to the opposite side. And the reason this is important is because we are going to use that energy to do so. And the reason we need to use that energy to do so because there's already lots of H+ here. There's already a ton there. Okay, so what we are doing is we are pumping H+ against its normal concentration gradient, where it is very highly concentrated on this side and relatively low concentrations on this side. We're taking these H+ and we are pumping them against where they naturally would want to flow. Okay, so if we correlate that to over on this picture with the dark orange and the light yellow, there's lots of H+ right here. And we're going to take whatever H+ is on this side, we're going to pump it to the other side. Okay? That's effectively what we're doing. So we are using all of that built up energy capital that we got in this electron transport chain. When these electrons started with some energy and went to a lower energy, they released that, and we're going to use it to pump those H pluses against their gradient, okay, against what they normally would want to. So we've set up a proton gradient out here. Okay, let me get a different color pen for this next one. I like that blue. So way out here, that free energy is utilized to make an area that has tons of H plus, okay, right out here. And that correlates to this space right out here in the diagram. All right? So that's what this diagram is meant to show you. Not necessarily, come on now. Not necessarily that you need to memorize all of these different protein names and stuff like that of the electron transport proteins. That doesn't matter, okay? I do want you to understand, though, that the electrons pass through there and they ultimately release energy. And by the time they get to this last one, they ultimately end up with oxygen to make water. Okay, that's where we get the vapor that is exhaled when we are done breathing, right? So let's look at this diagram on the right and see if we can describe this process a little bit more like storytelling, okay? So we can say that the NADHs and the FADH2s that were produced in many of the other processes are going to come to the electron transport chain and drop off their high energy electrons. So the NADHs are going to drop them off here, and the FADH2s are going to drop them off here. And so what we see is that as these electrons move their way through all of the proteins in the electron transport chain, they will release free energy. And as they release that free energy, these H pluses are pumped from the inner mitochondrial matrix, the yellow part, to this inner mitochondrial space, or this inner membrane space that we see here. Okay? And so this is going to yield tons of H plus out here, and I'm going to go ahead and put tons over here as well, tons of H plus over here. So we got tons of H plus in this little area, very acidic if you want to characterize it that way. Okay? So now we're with the electrons here at the final electron transport chain. These electrons need to go somewhere. One thing that if you continue to study biology, you will understand that electrons that bounce around and do nothing or have no place to go can be absolutely dangerous. I believe they're called free radicals, and they can cause a lot of havoc, okay? And so we need a place for these potentially dangerous electrons to go, and we're going to go ahead and put them with the only thing that wants them at this point. Really, at this point, the only thing that wants these kind of low-energy electrons is oxygen. It's the only thing electronegative enough to actually accept them, and once it accepts those electrons, so we take the electrons that we see here, combine them with oxygen, and we make water as a byproduct. Okay? Now, we still don't have any ATP yet, but what we do have is potential energy. That is what these tons of H pluses represent. The same way that the water reservoir on the top of the Hoover Dam represents potential energy in the fact that it can flow back down through the dam, spin a turbine, make energy, make electricity or whatever. That is the exact same thing that happens here. These H pluses will flow back down their natural concentration gradient, okay? Because they're very highly concentrated up here, they're going to want to flow back down with their gradient, right? But the only place that they can flow through is this molecule called ATP synthase, okay? And ATP synthase is a molecular turbine. It works almost identical to a turbine you'd see in a hydroelectric dam. And through the spinning of this turbine, it mechanically attaches the phosphate here to the ADP to make the ultimate product ATP. Okay? So that's how this entire process works. It seems more complicated than it really is. Okay? It seems like, why would they put all these steps together? I don't know. That's how it evolutionarily adapted, and that's a far greater question than I have time to answer here. Okay? But the idea is that these electrons bounce through the proteins of the electron transport chain. They will release free energy as they do this. That free energy is harnessed to pump H plus against its gradient to an area where it's already extremely highly concentrated. That H plus will flow back down through ATP synthase, spinning a turbine to make ATP. Kind of a cool process if you really think about it, okay? So let's get back into the presentation here and kind of finish up. I've already talked about the role of oxygen. Oxygen is in there, and it's in there because it's the only thing electronegative enough to accept the relatively low energy electrons at the end of the electron transport chain. Because at that point, they've given up all of their high energy. That high energy has been used for something else, and nobody really wants those low energy electrons except for little old oxygen. And when it does get that, it will actually grab onto those and make water from it. Okay? So let's go ahead and watch the short video. It's actually a really, really good one, depicting everything that happens.
Most of the energy harvested from organic molecules during glycolysis and the citric acid cycle is stored in NADH and FADH2. These molecules give up their high-energy electrons in the third phase of cellular respiration, and oxidative phosphorylation, where most of the cell's ATP fuel is produced.
The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondrion. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward highly electronegative oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and two hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed up hydrogen ions give up their energy when they diffuse through a special protein in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. This mode of ATP production is called oxidative phosphorylation because it is powered by the transfer of electrons to oxygen. Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by electron transport and oxidative phosphorylation, about 34 ATPs for every glucose consumed.
This illustration summarizes cellular respiration. Glycolysis and the citric acid cycle produce a small amount of ATP via substrate-level phosphorylation, but most of the cell's ATP is made via oxidative phosphorylation, when NADH and FADH2 produced in glycolysis and the citric acid cycle give up to oxygen the electrons obtained from organic molecules.
All right, so back to this we go. So let's take we're going to do a kind of a final stockpile of all of. Before we do that, they mentioned a couple of times ATP synthase, and so did I. And I want to make you aware that I have linked to this picture. If you click on it, it will take you to a YouTube video that talks about how ATP synthase actually spins like a turbine to create ATP. Okay, it gets into the nitty-gritty and some three-dimensional molecular models that show you exactly what to do. Now, you're not responsible for the information that's in that video or anything like that. But I just thought it's really interesting to kind of complete the picture of how ATP synthase actually does that. Because looking at a picture of stuff just flowing through this and then ATP is magically created doesn't give you the full story. And I like you to at least have options to check out the full story if you really want to. I will also try to link it in the classroom assignment. But let's go ahead and take a stock of what we've got. Now, One thing we have to try to understand is that we obviously have realized at this point that the NADHs and the FADH2s are responsible for ultimately creating ATP, right? Because they drop off their high energy electrons at the electron transport chain. And you can generally characterize the amount that each one produces within certain limits. So for instance, you can, rule of thumb, kind of say that each NADH is responsible for producing 3 ATPs. You can think of, rule of thumb, each FADH2 that is taken to the electron transport chain is responsible for making like 2 ATPs, okay? Those are kind of your rule of thumb, go-to numbers. If you ever see something that wants you to calculate how much ATP is made, and I'm thinking of like prior test questions that I've seen or used myself. Let's say you have the question wants to know you have 3 NADH. How many ATP were made? You would say 9, because you know that each NADH is responsible for making 3 ATPs. Okay, so the easiest way to remember the difference between these two is the fact that there's a 2 in FADH2. Okay, and there's not in NADH. So this 2 tells you how many ATPs it likes to make for FADH2. Now this last one, I throw it in for completion. Again, it's nothing that you have to memorize. Go with the rules of thumb that I mentioned before, but there's a certain amount of ATPs that are, it's almost like a range for the NADHs that were produced in glycolysis. So don't worry about this last statement. For what we need to know, for the most part, 3 ATPs are created from NADH and 2 from FADH2. Okay? So while you're watching this video, follow the slides that are going to be presented coming up and see if you can't just quickly do some calculations and figure out what the answer is to the question I'm asking. So what I want to know is how many ATPs we're getting total. So how many ATPs from the 2 NADHs we get from glycolysis? Pause it. You should have said 4 to 6, okay? You want to go rule of thumb, you could just say 6. You would have got it right, okay? But that talks to the range that we saw on the previous slide. The 8 NADHs from the mitochondrial matrix. This is the Krebs cycle and the link reaction. Okay, so that's a total of 8 NADHs. How many ATPs do we get? You should have said 24. Good. And then we get 2 FADH2s from the Krebs cycle. How many ATPs are they responsible for? You should have said 4. Okay, good. And so total ATPs from oxidative phosphorylation. Okay, that is the stuff that is going through the electron transport chain. Everything that is associated with NADH, okay, should be 32 to 34 if you add them all up. Okay, and if we take into account the ATPs from substrate level phosphorylation, so that would have been, I'll see if you can figure it out. I'll pause it. A total of 4. Right, so we had 2 from glycolysis and 2 from the Krebs cycle. We add it all up, and we get a grand total of 36 to 38 ATPs. That's where you get that famous number, which you have to memorize from the time you're like 4 years old, right? When you start to learn about cell respiration and how animals break down food and stuff like that. That is where you get all of your energy from. Okay, so I hope that the payoff was worth it. I hope you enjoyed watching the kind of icing on the cake discussing the electron transport chain, because to me this entire process is just absolutely fascinating when you take into account all of the different things that one glucose molecule goes through and you get this much energy. And then think about all the glucose molecules that are made in a given day or eaten in a given day and then broken down. And it gets to become these astronomical numbers, okay? So that is the process of cell respiration, and broken down into the 3 phases: glycolysis, Krebs cycle, and electron transport chain. I do hope you enjoyed it. And in the next video, we do need to discuss briefly what happens to this process in the absence of oxygen. Okay, because oxygen, I don't know if you noticed this, was a vitally important component to all of cell respiration to go off without a hitch and give you the maximum amount of energy that you can get. but sometimes that's not ideal, and sometimes you just don't have oxygen or enough oxygen to produce enough energy efficiently. And so the final video in Chapter 9 is going to address that situation.